Hello everyone, welcome back to our discussion on chromosomal mutations. We were discussing about the change in chromosome number, and in change in chromosome number, we discussed about how or what happens if the entire chromosome set changes. What happens when it is beyond or in addition to the normal one, like polyploidy, and what happens when it is less a set less than that of the normal one, which is haploidy and monoploidy. And now we are going to discuss about the change in the number of chromosomes. And in that, we are going to see the second type, which is the first type was aberrant euploidy, and the second type was aneuploidy. So aneuploidy is nothing but the change in parts of the chromosomal set. So it is not the entire chromosomal set, it is parts of the chromosomal sets. And we are going to discuss three types in aneuploidy, that is trisomy, monosomy, and nullisomy. So aneuploidy is the second major category of chromosomal aberrations in which the chromosome number is abnormal. It is not the chromosomal set, it is the number of chromosomes that is abnormal. An aneuploid individual is an individual organism whose chromosome number differs from the wild type by a part of the chromosome set. That's why we say that when you have aneuploidy with regard to a human chromosome, so the normal human chromosome number is 46, and that can change either to 47 or it can move to 45 or lesser or greater. Whatever it is, if the number changes rather than the entire set, and that is called as aneuploidy. An aneuploid can have a chromosome number either greater or smaller than that of the wild type. So the nomenclature is based on the number of copies of specific chromosome in the aneuploid state. For autosomes in diploid organisms, the aneuploid is designated as. So this is all about the uh, for autosomes in diploid organisms, the aneuploid can be written as 2n plus 1, which is called as the trisomy condition, which is one extra chromosome than the normal one. Then 2n minus 1, which is one less. So if it is a human chromosome set, trisomy is 47 and monosomy is 45. And you have another one which is 2n minus 2. So this too may be the loss of both the homologous chromosome. That means if for say if the 22nd chromosome is entirely, the two homologous chromosomes are lost, that is represented by 2n minus 2, and that is called as nullisomy. So three terms is what we discuss now in the case of diploid organisms. So for diploid organisms, I think for both autosomes as well as sex chromosomes, you use the same terminology as trisomy, monosomy, etc. So aneuploid is where the different types of aneuploid are where you have 2n plus 1. When there is one chromosome extra is called as trisomy. 2n minus 1 is called as monosomy. And if a pair, a homologous pair is being uh, not found in that particular, or if it is not there, it is called as nullisomy. And so this was the case with diploid organisms. In haploid, the term is disomic when you have n plus 1. So haploid, in the sense, it is not related to sperms or egg, it is related to haploid species. It is not also referred to drones. We call them as monoploids, but this particular haploids are those organisms which are entirely haploid. In them, if there is a plus one chromosome, and that is called as disomic. So this is a condition where you see these. So this can be compared. So this is a normal diploid that you see here with two sets of chromosome each. This is a normal condition. So you can see this is a normal condition here. What happens is if one of the chromosomes, so this is normal diploid, when one is lost, any one can be lost. If A is lost, it is monosomy. If B is lost, that is monosomy. C is lost, it is monosomy. So whatever the chromosome be, if one chromosome is lost, it is called as monosomy. If there is a gain of chromosome, can see 3A, 3B, 3C. If there is a gain of chromosomes, so the normal was 6, minus 1 was 5, plus 1 was 7. And if that can be of any of the chromosome, either it can be autosomes or it can be of the sex chromosomes. So whatever it be, it can be called as trisomy. And when in this normal case, if both this A is lost, they, that organism is having only BB and CC, such a condition that will be represented by 4 here, and that is called as nullisomy. So these are the different types with regard to this aneuploidy. So aneuploidy is just change in the chromosome number. So the different types, monosomy, which is 2 and minus 1, it is a missing of one copy of the chromosome. So in most diploid organisms, the absence of one chromosome from a pair, usually it is deleterious. In humans, monosomics usually die in utero, but there are many X chromosomal monosomics. Many X chromosomal monosomics also die in utero, but some are viable. The example of a viable X chromosomal monoploid is Turner syndrome. So, such syndromes or details of such syndromes we'll be discussing under the next the topic, which is human karyotype, etc. So, this is an example of a monosomy, which is Turner syndrome, which is a sex chromosomal monosomy. Next is trisomy. Trisomy is an extra copy of one chromosome. There is an imbalance from the trisomy condition and can result in abnormality or in some cases death. So there are examples of viable trisomy, and also in some cases there are trisomies which are fertile. So the examples of trisomy are Down syndrome and Klinefelter syndrome. So please note, usually in some textbooks or some students, usually they write an apostrophe s with this. So this is the correct terminology for this syndrome. It is Turner syndrome. It is not Turner's syndrome, and it is not Down's syndrome. It is just Down syndrome. And this is Klinefelter syndrome, not Klinefelter's syndrome. So these are the correct ones. If your textbooks also show an apostrophe s, please do correct it. It is Turner, Down, and Klinefelter. So now the cause, or what is the cause of this aneuploidy? We discussed the cause of aberrant euploidy. So now we are going to discuss the cause for aneuploidy. So the cause of aneuploidy is a process called as non-disjunction. So non-disjunction is nothing but it is a failure of either the homologous chromosome to separate during meiosis 1, or the sister chromatids to separate during meiosis 2 or meiosis 2, which is similar to mitosis, either meiosis 2 to separate during gametogenesis. So this particular, that is aneuploidy, usually result in or results from non-disjunction during gametogenesis, either formation of sperms or the formation of egg. So if this particular process of non-disjunction occurs during the formation of sperms or egg, this is called as this results in what is called as aneuploidy. So we'll take the definition once more. It is the failure of chromosomes to separate. So when they are not separated, so we know that they lie close together. There is crossover, there is recombination, etc. And after crossover recombination, the spindle fibers, they take one pair of one from the homologous pair to one pole and the other to the other pole. So that is what is called as equal, or which makes something which is called as a reduction, division, etc. So they move to opposite poles. So if that fails, what happens is uh, this particular homologous pair may move to one of the gametes. As a result, one will have a loss of a chromosome, the other will have a gain of a chromosome. So such conditions where there is a failure of either, in if it is in meiosis 1, it is the failure of the homologous chromosomes to separate, and if it is in meiosis 2, it is a failure of the sister chromatids to separate. So if this happens, that will result in what is called as uh, that is called as aneuploidy. Okay, so we'll see. We'll discuss this. So here you can see, though, so this is the first one, that is non-disjunction during the first division, or M1. So this is a homologous pair. We are talking about only one homologous pair, and consider that if we take this as humans for say, and we are considering that all other 22 pairs segregate normally. Only this particular pair or this particular homologous pair will be having non-disjunction. So what happens is, here the non-disjunction is going to happen in the during this is M1 non-disjunction, meiosis 1 non-disjunction, where homologous chromosomes fail to separate. So here you can see. So what happens normally is, this particular homolog, after crossing over etc. during anaphase 1, this homologous pair will move to one cell, this cell, and this brown should move to this cell. So what happens here is, there is a failure of them to separate. They do not separate. Uh, it can be either due to uh, the uh, the lack of formation of spindle fiber in one pole, or it can be due to, so there are several reasons that are postulated. So one reason is because of the lack of formation of a proper spindle fiber in one pole, so that it is not dragged, as a result that moves with the other one. Another thing is that the crossovers are needed for the bivalent pair until anaphase 1. If crossover fails for some reason, the first division non-disjunction occurs. So in meiosis 1, non-disjunction occurs usually because of lack of crossover. So that is the most probable reason for M1 non-disjunction. If crossover do not happen, that means they are not getting close together, they are not recombining, they do not cross over. In such cases, a bivalent is not formed, a tetrad is not formed properly. As a result, that can lead to a non-disjunction during meiosis 1. That can also be caused by an improper development of the spindle fiber. So two reasons are there, but the most probable reason which is being postulated for M1 non-disjunction is lack of crossing over. So if there is a lack of crossover, what happens is this particular condition. So the normal situation is like this. If M1 is normal, the situation should be like this, where one of the homologous pair is moving to one, and this is what is called as reduction division. But here you can see, when there is meiosis 1 non-disjunction, both the homologous pair is moving to one cell. So there is no reduction division happening. One cell is now with lack of that particular chromosome. So here you can see, here usually the gametes should have only one of the homologous pair. Okay, and that should be N. But here, two of the gametes, it is having now an extra chromosome of, or extra of this one. As a result, it is N plus 1. Okay, so this is N plus 1, and these are N minus 1s because they do not have a chromosome. So if I take an example of this, so if this is you will have something around, usually n should be 23. So that is a normal haploid number of a sperm or an egg. So this is gametogenesis. It can be sperm or egg. So the normal number is 23. So here what you have is 24. Okay, rather than the normal 23, there is 24, and here you have 22 chromosomes rather than 23. Okay, so we'll take a hypothetical situation. I am going to consider these as eggs, and consider that these eggs are fertilized. All these eggs are going to be fertilized by a normal sperm. Means the sperm will be normal with n number of chromosomes. Okay, so these all the sperms are containing n, that is all are normal with n chromosomes in it. Okay, so what will be the situation now? So normally, if this is n and the egg is n, you should get n zygote. If it is normal, you get a zygote which is 2n. When you have n plus n, which is n, the other one, that is n of egg and n of sperm. Here the situation will be, this one will be 2n plus 1, and this will be 2n minus 1. As a result, what happens here is, so have taken a situation where these are fertilized by normal sperm. So these are eggs. And here what happens is, these two will be the condition which is called as trisomy. And this is the condition which is called as, these two will result in the condition which is called as monosomy. So if non-disjunction happens in M1, all the gametes are having some problem, and all the even though the male gamete is normal, all zygotes will be abnormal. All the zygotes which is formed from that particular couple will be abnormal. That is because of this non-disjunction that is happening during meiosis 1. So no fertile or no viable offsprings are produced if non-disjunction happens in meiosis 1. So now we are going to take the condition what happens if non-disjunction happens in meiosis 2. So if you consider that, we are considering that M1 will be normal. So that is this M1 is normal, this is reduction division, one has moved here and the other has moved there. And we are taking a condition where this two cells, they show normal mitosis or normal M1. So this is the normal condition. So each one will receive each of the sister chromatids. So this is normal meiosis 2, where the sister chromatids are separated into two cells. So here, this particular chromosome, there is non-disjunction, where you can see the two sister chromatids are moving on to one cell, so that the other will lack. So this is n plus 1 and this is n minus 1. So here also we'll take the condition where they are fertilized by, all of them are fertilized by normal sperms. So if in this case, because 50 percentage is normal gamete, here you will have 2n plus 1 trisomy, 2n plus 1 trisomy. This is 2n minus 1 monosomy, and these two are normal or will generate normal zygotes, which is 2n. So this is the condition, that is, if meiosis or if non-disjunction is happening during M2 or meiosis 2 or during anaphase 2, there is a possibility that 50 percentage of the gamete formed will be normal and 50 percentage will be abnormal. So for such individuals where non-disjunction happens in meiosis 2, there is a possibility that they can have normal children, because half of the gametes are formed as normal. So this is what is non-disjunction. And here, if you check, I have taken as non-disjunction which is happening in eggs and not in sperms. So this I mentioned this as an example deliberately, because non-disjunction usually happens in egg, and rarely, or it is a very, very less chance, less than 0.001 percentage in sperms or during spermatogenesis. So the reason is that uh, spermatogenesis is a process which starts in the body of a male inside the testes. It starts at puberty, and it is a non-stop process. There is no arrest or there is no stoppage from puberty till that this particular process of spermatogenesis happens. So spermatogenesis happens, but as aging occurs, the rate or the number of sperms produced will be less, but the process will be still functional if that person is normal without any other disease condition, etc. So if you consider a normal person which is normally aging without other much complications, etc., this process of spermatogenesis inside a male's body, inside the testis, it will happen with a limited rate till death. But in females, the situation is different. In females, what happens is that in females, the process of oogenesis begins even before that particular female is born. That means the process starts inside the womb of the mother if that embryo is female. or the fetus is female after the ova, or the after the ovary is formed itself. The process of oogenesis is initiated, and oogenesis means it is meiosis. So it will be initiated, and after oogenesis is initiated in the oogonial cells inside the ovary, it will be arrested. Okay, arrested in the sense it will start meiosis or gametogenesis, and the first arrest happens before the baby or before the particular female child. This happens for every female, even before the child is born. It starts gametogenesis or meiosis or oogenesis, and it is arrested at prophase 1. So that is the first phase. So it just starts meiosis, and it is arrested. All the oogonial cells, they are arrested at the prophase 1, and that arrest uh, will be extended till the uh, puberty or the first menstrual cycle. So what happens is, in the first menstrual cycle only, if for say there are around for say something around in 1 lakh oogonial cells inside both the ovaries. There are two ovaries, so around 1 lakh oogonial cells are there. In the first one menstrual cycle, 20 or so oogonial cells will get a chance to resume meiosis. So they resume meiosis from prophase 1. So that is in the first cycle. Only 20 will get a chance. So 20 will start this meiosis in both the ovary. They will continue with prophase 1, then metaphase 1 and aphase 1. Then first polar body is formed. Then they enter this particular phase, which is the M2, and the second arrest occurs at ovulation. That is, at ovulation, that particular oogonial cells are arrested at metaphase 2. That means oogenesis is not complete when the ova is ovulated. It is, or it will be completed only if that oocyte is being fertilized by a sperm. If not fertilized, that ova will be removed through the menstrual flow from a female's body in an arrested state without the completion of oogenesis. So oogenesis for an oocyte is complete only if it is fertilized by a sperm and a zygote is formed, or else it is arrested at the metaphase 2 of meiosis 2. I said there is, so this is the normal procedure. So after puberty, every month in every menstrual cycle, such 20 oogonial cells will start this meiosis, but only one will get a chance to reach metaphase 2. All the other out of the 20, one or two will be ovulated depending upon the genetic makeup of that particular female. One or two will be ovulated, and what happens is all the others which have started this particular process of meiosis, they will be degenerated inside the ovary. Degenerate inside the ovary, and such degeneration can be seen throughout the female's life, and that degeneration of oogonial cells inside the ovary is called as the process, which is called as atresia. So atresia is the degeneration of the oogonial cells inside the ovary, and this happens in every cycle. So what is the relation with this to this aneuploidy? So the relationship is that usually this aneuploid cases like Down syndrome or Klinefelter or whatever it be, monoploidy or triploidy, such syndromes are usually associated with the age of the mother, not with the age of the father. Usually it is associated with the age of the mother, because if a particular female is consuming after the age of 45, there are more chances of this particular uh, aneuploid individual or aneuploid offsprings to form. So there are uh, uh, genetic predisposition also. So in normal females, that means without a genetic predisposition to this aneuploidy. So if a particular family is having a predisposition or genetic predisposition to aneuploidy, in the whatever the age be, age does not matter. All the ones which is the individuals they consume, that might be aneuploid from the if that particular female is genetically predisposed. If she is getting pregnant at the age of 20 also, if there is a predisposition, she can have aneuploid ones. But in normal females, if that particular female is trying to get a baby, or she is getting pregnant after 45 years or after 50 years, there are more chances, even though she is normal, without a genetic predisposition, she is having a chance or a greater chance for aneuploid eggs or aneuploid offsprings to be produced. And that is because we said that the oogonial cell started the meiosis, the process of oogenesis, even before the child was born. Okay, so they were arrested in that particular point, and these oogonial cells resume meiosis only in each and every cycle. So a group will be, so if a particular female is getting pregnant after the age of 45 or 50, just assume that that particular oogonial cell was in arrest. That particular oogonial cell which was fertilized, or which is going to form that offspring, or which is going to be ovulated in that particular cycle of 50 years or so. What happens is that was at arrest for 45 or 50 years. That has started oogenesis in the ovary, and that was arrested, and it is now getting a chance at the age of 45 or 50. So what happens is there will be the processes like the spindle fiber formation, the process of crossover, etc. There might be many errors which are happening because the system has now weakened, because it started 45 years and it was waiting for this 45 to 50 years. So the process of spindle fiber formation or the process of crossing over, etc., might have errors, and that will lead to aneuploid gametes being formed, and that will result in the formation of aneuploid offsprings. And this is the reason why usually such aneuploid syndromes, whether it be monosomy, whether it be trisomy, that is usually related to the age of the mother and not with the age of the father. So mm, uh, the uh, the syndromes or disorders which are associated with abnormality in sperm is very rare, very rare. Okay, the reason is very simple. The reason is that, so the process of spermatogenesis can also lead to errors, because it is a very fast process. It is a very fast process happening in a male after puberty. It just goes on happening, or it is a continuous process, and it is a very fast process. There is no errors, etc. So what happens is, in every spermatogenesis, okay, what happens is 1 to 2 percentage of the sperms which is formed will be abnormal. So usually, you know, the structure of the sperm, it is having a head and a tail. And in every spermatogenesis, for say millions of sperms are produced by males. In that in that millions, 1 or 2 percentage is a very less number. 1 or 2 percentage will be abnormal. Abnormal in the sense, some sperms will have two heads, some will have two tails. Okay, such abnormalities are there in sperms, but that abnormality will not be reflected in the offsprings. The reason is very simple, and the reason is that only properly formed and healthy sperms reach the egg. Even if this abnormal sperms are deposited in the vagina along with the normal one, abnormal ones will not reach the egg and fertilize it. Only normal sperms can reach the egg and fertilize it. And that is the reason why the syndromes or disorders which is associated with errors in spermatogenesis are very less compared to that with the oogenesis. So this is the cause of aneuploidy, and that is what is called as non-disjunction, etc. So in the next session, we'll be discussing the second type of mutation, which is gene mutation or point mutation. Thanks for hearing.